So next, I want to welcome Oluwatife Irewolede from Helix Biogen Institute in Nigeria. He'll be talking about Immunoinformatics approach in designing mRNA vaccine against SARS-CoV-2 variants.
Good afternoon. Good afternoon. Please, hope you can hear me.
Yes, we can hear you and we can see your screen. However, I think you need to be in presenter mode because right now we can see all of your slides. Oh, that's perfect. Thank you.
So I am Oluwatife Irewolede by name. I am a research assistant at Helix Biogen Institute, Nigeria. I will be presenting this research work titled Immunoinformatics Approach in designing mRNA vaccine against SARS-CoV-2 variants. The proliferation of COVID-19, which has led to over 1 million deaths worldwide and also caused wreckage on the economy of several nations of the world ever since its eruption in December 1st, 2019, has caused a lot of concern for many public health practitioners across the globe. And as of the 5th of August 2021, according to the African Renewal, it was reported that over 6,000 deaths were recorded within a week, with 19% increase in the confirmed cases of COVID-19 at almost 300,000. And due to the recurrent mutations that occur across the genomes of this deadly microorganism, several variants of SARS-CoV-2 has emerged across continental boundaries. And according to reports, it has been said that about 6,000 data variants was recorded along the shores of more than 20 countries in Africa, with South Africa scooping the greatest percentage of this unpleasant situation. And it is by this effect that we subjected to carry out this research, most especially within the African populations, where there are limited technologies and provisions to combat the virus. Though several methods have been devised by many researchers to develop an effective vaccines to prevent the spread of the disease. Though these methods have successfully provided a durable protection, but nonetheless, according to reports, mRNA vaccines is said to hold a great promise for the future, and as it has been proven to show several advantages over other methods. And besides the safety and the potency of this particular vaccine, mRNA vaccine has enormous flexibility in its design and in its production. And this particular vaccine, it functions by first utilizing the host cell genome by translating the mRNA into corresponding antigen, thereby mimicking the viral infection, and from there it elicits an humoral and cellular immune response. So this is the methodology of the research work that was carried out. This methodology is divided into 13 different sections. The first of it is the retrieval of SARS-CoV-2 sequences. We make use of the GISA database to retrieve our sequence, and the data that was used in this particular project research work was targeted towards 5 African countries, namely Angola, Botswana, Mozambique, Lesotho, and Namibia. And 189 sequence, the whole genome sequence of SARS-CoV-2 was retrieved that was submitted to the GISA database between December 1st, 2020 and 5th of March, 2022. 2021 was retrieved and was analyzed in this particular. After the retrieval of sequence, we later on carry out some level of evaluations. The first of it is we went ahead to predict for the prediction and the evaluation of CTL epitopes. From there, we also proceed to HTL, prediction of HTL and evaluation. From there, we went to predict for the prediction and evaluation of the linear basis. From there, we carried out a lot of evaluation on each of the epitopes, and from there, we're able to arrive. We also carried out population coverage, just to show the effectiveness of our vaccine construct, how it can the effectiveness of the vaccine construct. Also, in order for us to be able to target mutations that occur, so we were able, using in-house R script, we're able to to randomly select 100 sequences of SARS-CoV-2, and we submitted to multiple sequence alignments. After that, we were able to construct our vaccine, and after the vaccine construction, we were able to do a kind of prediction such as antigenicity, allergenicity, toxicity, and physicochemical properties of the vaccine construct. After the prediction, we went ahead to predict the structure, and we also assess and validate the structure that was predicted. From there, we went ahead to do the confirmation B-cells prediction, and from there we do the molecular docking with Toll-like receptors. From there we go forward to do our molecular dynamics. So this is the result and the discussion of our work. Unlike DNA vaccines, SARS-CoV-2, being an RNA vaccine, has the propensity of undergoing recurrent mutations. And in order for us to validate that the epitope that was selected for this particular vaccine construct has not undergone mutation, so we make use of, we subjected it to multiple sequence alignments, and from here we were able to get a fantastic, interestingly, we were able to realize that no mutations was observed within the selected 7 epitopes in the 100 SARS-CoV-2 sequence that we subjected to multiple sequence alignments. And here we have our vaccine construct. Here now, before we make use of ECIAC and CIMIA procedure in constructing our vaccine. So the vaccine comprises of 7 epitopes, we have 1 HTL epitopes, we have 1 LBL epitopes with the linear B cells, we have 5 CTL epitopes. And we also incorporated into our vaccine construct the highly immunogenic adjuvants. And in this case, we make use of CD40 ligands. And this particular ligands we involve the involvement of this particular adjuvant in this study was considered owing to its inherent efficiency to stimulate the professional antigen presenting cells, which could invariably lead to the induction of immune response molecule. Also, we also included MH1 target domain, and the function of this particular domain is just to direct the CTL epitopes to MH1 compartment of the endoplasmic reticulum. Also, we included signal peptide, and an example of the signal peptide that we used was the tissue plasminogen anti-activator, and the function of this is just to secrete translated epitopes which are needed to move out of the cell. So that due to the shortcomings that were encountered during the production of mRNA, notably the stability and delivery of mRNA, we included 5' UTR and 3' UTR in our vaccine construct. This is just to ensure the sufficient production of antigen and efficacious vaccination of the host cell. And the 5' UTR functions for efficient protein production, while that of 3' with its associated poly-A functions to determine the stability of mRNA and increase protein translation. So this just the construct that we have, and at my right hand side, just by it, we have a map here, and this particular result is just telling us the result of the population coverage that was predicted for the vaccine construct. So the combined coverage of the CTL and HTL epitopes was assessed just to predict the effectiveness, just like I said before, of the vaccine construct within the study area. The epitopes show a good population coverage of 77.22% in average, and we also have a high degree of coverage were predicted for all the regions in this study area, thus indicating the possibility of the vaccine construct to stimulate an immunological reaction within the population of the study area. Now, this is the result for the physicochemical properties and the secondary structure prediction of our vaccine construct. So now the physicochemical properties of this vaccine construct proves its ability to be a good potential candidate for a vaccine. If you can see, the molecular weight of the vaccine is 223.1 kilodalton, and which says that it is an average. The result we got from the molecular weight is far higher than the normal threshold of 110 kilodalton, and this one give us give us the assurance that this vaccine is going to be a good candidate. Also, we have the theoretical p-high of 8.69, which shows that it is slightly basic. Also, we have the instability index to be 48.78, which shows that it is unstable. Normally the threshold value for instability index supposed to be 40, but because of the nature of the vaccine construct that we are designing, so it's this particular result that we got here is really confirm the problem that is being and this problem of instability that is being that befell the production of mRNA. So now we also have the aliphatic index, which is 82.13. This shows that our vaccine construct is thermostable, and the grand average of hydroplasticity is minus 0.296, showing that by the time the vaccine after its expression has the capacity to be able to mix with the soluble substance in the body system. And from our prediction, from physical chemical prediction, we were able to realize that our vaccine is antigenic, is non-allergenic, and is not toxic. And this at the right hand here, we have the result for our secondary prediction. As we all know that in the development of vaccine, having the knowledge of both the secondary and tertiary structure of the target protein is crucial, as it provides a better understanding of the constructed vaccine candidate. So the analysis of our vaccine secondary structure revealed that the protein contains merely 33.38% coil. Now, the figure 6A is just showing the representation of our 3D structure of the vaccine construct, and the 6B is just showing the refined vaccine 3D structure by using the galaxy refined web. Also, the figure 6C is just giving us a clue about the vaccine 3D structure validation, which was carried out using PROSA web. So the Z-score of this refined model is minus 7.6, which is lying inside the score range. Also, the figure 6D is also giving us the information about the residue score plot by PROSA web to check the local model qualities and negative values, suggesting that the no erroneous part of the model structure was recorded, as you can all see here. And also for the last figure that we have, a 6E, is also showing the Ramachandran plot assessment of the multi-epitope vaccine construct. And here, the result of the Ramachandran plot indicate that 97.8% of the residue were initiated in the favor region, while 1.3% is indicated in the allow region, with less residue of 0.4% in the outlier region, thus suggesting that the quality of the whole model is acceptable. On the right hand side, we also have the result of our molecular docking. As we all know that the molecular docking is a very important tools that are used in bioinformatics tools that are used in carrying out the evaluation, the stability, and the stability and orientation of our ligand and receptor value. So now from here, the blue color is showing our vaccine, while the other structure here is showing it indicates our TLR, and here we make use of TLR4. The essence of our TLR4, of using TLR4, is that TLR4 is just TLR4 is an important human protein for pathogen recognition and immune responses. So this is just the result gotten from our molecular dynamics simulation. This is just to show this particular result is just to show the stability movement of our vaccine TLR based of current pandemic challenges, which has ravaged lives. The development of vaccine is an urgent need for the world. The result generated from the research revealed the vaccine construct to be a promising therapeutic agent in combating a deadly virus, and this will only be possible following further validation of its performance in vitro and in vivo through coordinated works by the relevant parties and stakeholders in the health sector. Also, an acknowledgement, I appreciate the staff and management of Helix Biogem Institute, Ogbomoso, for the technical support throughout the course of this work. Also, I appreciate all the BSI Conference Committee for giving me the platform to present this work. Thank you for listening.
Thank you so much for that fantastic presentation. We just have a couple questions if you don't mind. So the first one is, why did you choose 5 CTL segments and not more?
And we realized that this city, they are part of the city that let me say these particular countries are part of the countries that are underdeveloped in Africa per se. And we just try to like check out if this particular countries has the data of SARS-CoV-2 in the GISAID. And fantastically, out of the 5 countries that we choose as our study area, 3 of the countries has its data, SARS-CoV-2 data deposited on the GISAID database. While 2, the 3 countries that has the data deposited on the GISAID database are the Angola, Mozambique, and Botswana. The other 2, Namibia and Lesotho, they don't have the data of SARS-CoV-2 as at the time when we were working on the work on carrying out the research. They don't have the data of SARS-CoV-2 deposited on GISAID database.
Thank you. And also, how will you test antigenicity beyond predictability?
What do you say, ma'am?
Sorry, how will you test the antigenicity beyond predictability?
Okay, we make use of two servers, online tools, bioinformatics tools to carry out our antigenicity. The first one is Antigen Pro, and the threshold value for Antigen Pro for prediction with Antigen Pro is 0.8. And from the result we got, the result for Antigen Pro says that okay, we have 0.733 because the large we have a large volume of the primary source of our vaccine, so we have to split it into 2. After prediction, we now added the score together. Also, we also predict with the vaccine, and the threshold value that was set for our prediction was 0.5. So anything above 0.5 and above is said to be antigenic.
Sorry, just to return to the CTL question. So the question was referring to the CTL semester in the construct, how you had 5 CTL constructs.
Yes, after looking at the methodology we were able to predict 36 epitopes, CTL epitopes was predicted, and we now later subjected each of the epitopes to further analysis. One of it is the immunogenicity, just to screen down the volume of our CTL epitopes that we got. At the end of the day, we were able to arrive at 5 that passed the different evaluations that we subjected them to. So that is the reason why we have to arrive at 5. Same thing as HTL and LBL too, just to make sure that we are able to pick a good epitopes for our vaccine constructs.
Great, thank you so much for your time.
Yes, ma'am. Thank you.